Parkinson's disease affects nearly 1 million Americans, but treatments have been limited to managing symptoms rather than slowing the disease itself. Now, a clinical stage biotech company is using an AI-powered drug discovery platform to take on one of neuroscience's greatest challenges. Its lead candidate is already showing signs that disease modification may be within reach. Hi, everyone. I'm David Williams, president of strategy consulting firm Health Business Group and host of the Health Biz podcast, where I interview top healthcare leaders about their lives and careers. My guest today is Gene Mack, CEO of Gain Therapeutics. Under Gene's leadership, Gain is advancing a potentially groundbreaking disease-modifying therapy for Parkinson's disease while deploying its Mellin AI ML drug discovery platform to unlock additional treatments. Gene, welcome to the Health Biz podcast.
Thanks, David. Thanks for having us.
Now, I understand you joined Gain around 2024 as the CFO and then quickly switched over to running the company. And I think you said at that point you really didn't want anybody else in charge. So I'm wondering, what was it about Gain and the science and mission that made you so certain about that?
The company wanted to take a more focused approach to its capital markets strategy and financing. And in the process of bringing me on board, they decided more and more responsibility. It's not something I'd never. I would say no one comes out of the womb wanting to be the CEO of a biotechnology company. I made it well known that my job was supposed to be second base for the Yankees, but I couldn't get there. And so, when I was joining Gain, the role, they said the role may be in flux. They asked me about the CEO, would I ever see myself as a CEO? And I kind of said no. I liked finance. I liked my lane. I liked what I did. I had a lot of expertise. For better, I don't love, dealing with a board is a separate job. You know what I mean? So, but they're lovely and they've been so supportive and they really made it a very, really sort of gratifying experience. And I feel like I've grown and I've learned a lot in the last 2 years. So I'm glad it all played out the way it did. And yeah, I still believe that. It got to the point where, I was an interim CEO for about 6 months prior to them making it permanent. And by the end of that 6 months, I was really nervous that they would go in a different direction because by that point, you're starting to wrap your arms around everything that's problematic, and you sort of see your way clear to a path. And you're like, well, we bring in somebody new. What does that do to all the things that we think we can have rolling? So ultimately, I think it all played out the way it should have.
Parkinson's patients and families are well aware of the limitations of current treatments. It's really just about managing symptoms, at least what I've seen thus far. And my belief is that Gain's approach is pretty different. Can you explain what it is that the drug does and why you think it could be the first disease-modifying therapy for Parkinson's?
Yeah, so you're absolutely right. For decades now, the only relief we've been able to offer to Parkinson's patients has been some kind of mask over their symptoms, and that wears off over time. And the pathology in the disease progresses. So what do you do to address that? Well, you get to the underlying biology and you try to correct on a molecular level what's happening to create these symptoms. And we believe that GT-02287 is going to be among the first, if not the first, disease-modifying therapeutics based on the data that we've seen so far, the preclinical efficacy that we've seen, or the preclinical mechanism that we've seen this drug work through. Suggests disease modification at every level we have tested it at, including what we think is, the phase 1B in patients now, Parkinson's, where we're seeing biomarker evidence. These are the actual biological indicators of how the disease is progressing. We're seeing what we believe is some real impact there in the presence of GT-02287. And we believe it's linking to a clinical benefit. It's early days in this study still, and we have low numbers of patients, but the signals are impossible to ignore. And that's what's really exciting about what Gain's doing now. We've just recently presented data at a global conference in Copenhagen called the ADPD conference. Data was very well received there, and we think we've given the field a lot to think about now in terms of how what we're seeing in the biomarkers, and hopefully they're going to do work that's similar to ours and help us understand it better based on the drug development that they're doing.
Great. So let's just go a little bit deeper in terms of how things work. And I'm just not an expert, but just looking at your materials and seeing that it appears that your drug is targeting a misfolded enzyme and works through an, I think, pronounced allosteric mechanism. It's not binding to the active site. Why does that matter? And what makes this kind of allosteric modulation potentially a better approach? So not just you're getting the clinical evidence, but why is it that you think that may be an underlying better way to go?
Yeah, and that question is central to what we're trying to achieve here too. So the allosteric binding, what does that mean? So there's different ways to bind a target protein or enzyme. Typically, if you bind it at the active site, you're looking to probably try to inhibit that particular enzyme. Maybe it's overactive or it's misfunctioning in a hyperactivity way, right? So you want to disable some of that enzyme. Sometimes you can bind to the active site and actually enhance the activity too. It all depends on the binding kinetics and what's happening in that particular area of the molecule and how you're impacting it based on that binding. Now, what we do is we move mostly further away from the active site. We're looking to stabilize the enzyme. So if you remember from high school biology, the lock and key mechanism for proteins and enzymes, their function is determined mainly by their conformational shape. So if that gets disrupted either through genetic mutation or some sort of post-translational modifications to the protein, so after it's transcribed and it's sitting in the cell, if something comes along and disrupts it. So what can you do in order to preserve the function of an enzyme that might be genetically malformed or under assault some other way? Well, so we bind to an allosteric pocket on this enzyme, stabilize the conformational shape of the protein, and chaperone that enzyme through its traffic pattern in the cell. And the particular target of GT02287 is an enzyme called glucocerebrosidase. It's a mouthful. We shorten it to GCase. GCase is a very well-defined, fairly well understood enzyme that is prominent in the pathophysiology of Gaucher's in the periphery mainly. So that's another disease where this particular enzyme is not working properly. But in Parkinson's patients, it also plays a role too, particularly if you have what's called a GBA1 mutation in the gene that encodes for glucocerebrosidase. If you're carrying a mutation in that gene, your enzyme function is going to be naturally lower than a normal individual or a person who has normal levels of GCase activity. So we're trying to enhance that protein activity, that enzymatic activity in Parkinson's patients, because that enzyme is responsible for clearing out toxic substrates that build up in the cell and create those symptoms over time.
So you're talking about a genetic mutation, and my understanding is that the drug was originally designed for that subset of Parkinson's patients that had that mutation. But also, I think maybe some of the excitement about the early data is saying, well, maybe it's actually a broader population that's relevant. Is that the case, and is that a big deal? If so,
yeah. So the prevalence of the gene mutation, the GBA1 mutation, is about 10 to 15% globally. Now it depends regionally and ethnically. In a lot of cases, there's some distribution of that percent. It's not. For example, for whatever reasons, Ashkenazi Jews have a much higher prevalence of a GBA1 mutation than other races do. There's a population of Northern Africans that also carry a higher prevalence of it as well. Not well understood why that is, but so some of that is so there are some certain populations that express this mutation more than others. But overall, if you look at all the literature, you find it's about 10 to 15% of the patients, which would maybe make it fall into sort of an orphan category. We're agnostic to orphan or otherwise. What we don't want to do is leave any Parkinson's patients behind. If you carry the mutation, our hypothesis with GT02287 is you may respond better to our intervention. However, all patients with Parkinson's can benefit from an elevation of their GCase activity because there's clearly a buildup of toxic substrate genetically driven from a genetic mutation or otherwise. There's other things that can happen to proteins too that are not related to a genetic mutation that may be occurring more in Parkinson's patients, right? So outside influences and toxins, things that get into the cellular microenvironment, like pesticides, those can drive Parkinson's. And even though GCase is not mutated in that particular individual, they may benefit from a superdose of GCase, right? So we elevate their GCase activity to overcome the other onslaught. And so we think that we can cover all Parkinson's patients with potentially differential responses among patients who have a genetic mutation, those that are, idiopathic instead, which is not a very ill-defined way. There's no real understanding of how the Parkinson, there might be another genetic mutation or some other cause that we don't know about. But we think that both populations can benefit from GT02287. So for now, we're enrolling everybody to see if we can tease out how these different patient phenotypes, how they present, and they all present very differently, how they all react to the drug, so we can better understand how to address patients.
So it sounds like a very interesting program. You also have a broader Magellan platform, and I've seen it described as a physics-based AI, ML, drug discovery engine. Now, obviously, AI is a place that we want to be. At the moment, there's a lot of hype about it in drug development. I'm not saying it's for real, but I'm wondering, is Magellan for real? And is there something about this approach that you're taking that surpasses what a traditional approach would be able to accomplish?
Well, yeah, so it was an earlier iteration of Magellan that actually led to GT02287. So we've actually been refining it and improving it along the way. We've actually, the backup series of compounds, the GT02287, were also developed using Magellan. So we find it to be a very efficient platform. The thing about AI in drug development, at least the way we're deploying it, right, is that there are, in order to find novel binding sites on proteins, you have to understand the binding and unbinding kinetics that a small molecule has to achieve in order to get into that binding pocket. And then you have to figure out whether or not something like that can be constructed if it's not already in the public domain. In other words, we're also trying to expand development with AI beyond what's in the public domain to come up with molecules that maybe never have been formulated before. Well, that's a lot of calculation, and some of which that takes 10 minutes to conduct, right? You have to do that over billions and billions of iterations. You can see the time, right? So our sort of approach to this is to use proprietary physics-based calculations that are automated and can do all that in milliseconds. And so by constantly refining that, get through those binding kinetic iterations much faster and arrive at a molecule sooner. Now, the thing about AI is we can pump out theoretical molecules all day long. At some point, you got to get them into you have to pull up stakes and validate the platform through clinical development, and that's a much slower process that AI is going to. We're going to need to put a pin in how we deal with the human aspect of if there's any way that we can bypass some of the human testing that's done by using AI. I think that's going to be a heavy lift. But at some point you have to go through that very well defined much more slow moving path, and that's what we're doing now. So Magellan has gotten us to this point, but now we're a clinical stage biopharmaceutical company developing clinical assets, and we need to focus on those. Magellan continues to be refined. We believe there's a lot of value there. We can always go back to it for a new molecule, but right now our resources are tied up in what we have in the clinic.
Got it. We're getting to the point where with the help of AI, you can do in milliseconds, generate enough ideas that would take a lifetime to be able to actually test out.
Yeah, that's, we have to, exactly.
So how do you go from, I get the shift, and it's a big milestone to get to be a clinical stage company, and especially not just clinical stage, but also have something that's interesting and, creating data and serving a population that may make the market size 5 times what you originally thought. So that's all good. How do you think about then, let's say, taking the next step and using the platform and its new, AI based approach and use it to, generate a pipeline versus being a one drug company, as exciting as that may be by itself?
Yeah, so again, it comes down to target identification, going into the biology, looking for, across diseases, looking for those bad actors. In our case, it's proteins that need help. are sick. And so which one of those proteins do we want to put through the model to see if we can come up with a novel molecule that we can then test in the clinic? But once you do that, there's a whole battery of IND. You have to do a whole bunch of preclinical testing on it. So it's not like you just get right into the human being as well. So building a pipeline with AI is still going to be very, very labor intensive. You can get to at least the way we're deploying AI, we're getting to molecules that way. We're not able to prosecute their early preclinical development with them. Again, you're still, you could be up to 10 years away from a drug once AI is done with its lift, right? I mean, it can still take that long. Now, hopefully it's more like 3 to 5. That's more or less what we're trying to achieve based on when we initiated the clinical development. But there was a lot of work that went on behind that, in front of that had nothing to do with AI.
When we started out this conversation, you talked about enjoying being on the finance side and not having to deal with some of the stuff that the CEO may do. Now, not that the CEO doesn't have to deal with this, but the capital markets have been pretty tough for biotech lately. I guess that's an understatement. But how do you think about where we are at the moment that would explain why it's so tough, and how do you navigate in that type of environment?
Yeah, I think there's a lot to say there. I've been in this sector and around this sector for 30 years now. I can tell you this has been going back to when things got difficult in biotech in terms of access to capital. That goes back to probably starting in the middle of 2021 and getting really bad towards the end of 2021, and then into 2022 it was completely frozen. This that has taken way longer than anybody had thought it would to kind of wash through a typical biotech bear market. And there's bubbles. I can recall off the top of my head at least 3 or 4 very significant ones in the sector over the course of my career, with little ones in between. There's usually a much faster. The difference with this one is the snapback has been gruelingly, grueling. So, I mean, there are signs over the last 6 months or so that things are improving, but it is a very, very slow walk back to days where capital was accessed on a much more rational basis than it is today. What's driving that? Well, it's a combination of things. We have a strange interest rate environment, an administration that's very difficult to predict, and a global crisis that seem to kind of wax and wane on a daily, weekly basis. That's the kind of uncertainty that the market, it's hard enough to predict the things that are already difficult to predict. And then, layering on top of that a bunch of geopolitical and macroeconomic, challenges. And you've got no one wants to invest in a company that's going to be illiquid for 3 or 4 years while they figure out if the drug works in the clinic. That's all this is. It's not even a testament to whether or not, biotech is doing a good job of developing drugs. Biotech has not skipped a beat. We're still pumping out, new drugs and new drug applications. But the access to capital for them, it's the investor's appetite for risk that's changed, not our ability to deliver. And their appetite for risk right now is just really not very big. It is very small. And they want de-risked opportunities. And that's why we spend so much time in our phase 1B looking at the biomarkers. The biomarkers are meant to give people comfort, the field, scientists, clinicians, investigators, investors. It's meant to give them comfort that we're on the right track, and we have a much better shot being successful in a phase 2 study, which is larger and going to be powered for statistical significance, on the back of signals that we know that we have a high degree of confidence are real in a phase 1B study. So we develop these objectives for early stage studies, so we know we can de-risk the later stage studies, and we think we've done that successfully to address the market's requirement. Normally, we went back 5 years ago. This drug, we wouldn't have even done a phase 1B study for signal. We would have gone straight once this was healthy. In the healthy volunteer study, the study that we conducted prior to this phase 1B was a phase 1A in healthy volunteers. You have to first show it's healthy and it's safe in healthies before you can move into a more fragile population like patients. In the healthy volunteer study, we saw target engagement. We saw an increase in GCase activity in those healthy volunteers. That's one of the reasons why also we moved out of the GBA1 population. If we can elevate enzyme activity in healthy volunteers, let's go after all the Parkinson's patients first. And then if we unfortunately have to slim down the population, then we'll do that. And let me be real clear here. This is not a commercial decision. The mission at Gain is to improve patient outcomes through the application of small molecule protein enhancers. That's our mission. We can get to large commercial success, whether this is an orphan drug or it serves a much wider population.
No, that's good. I was wondering what you were going to say about how you discovered that it could go beyond the mutation, because you wouldn't have discovered that in phase 1B if you just recruited people with mutations. So there was this phase 1 signal phase 1A signal that gave you that. So yeah.
Yeah. So out of curiosity and just out of out of intellectual curiosity, we looked at because there's no reason to believe that we would activate GCase. These things, enzymes work in a homeostatic environment. So if you even if you transiently increase a particular enzyme, so there's probably some other feedback mechanism to compensate for that and bring it back down through some other mechanism. We saw it in the healthy volunteers, so that we changed our view on a target population. Say let's bring in all comers first and see how they respond. It can also help the field understand more about GBA1 Parkinson's versus idiopathic Parkinson. We owe the field that as well. So we want to put this into all comers right now and really kind of look individually based on patient characteristics how they respond.
Great. So let's talk about that upcoming phase 2 trial, which you mentioned, of course, is going to be bigger, and I understand it's around 100 to 200 patients, which should be a little easier, I guess, to enroll if you don't have to focus just on the mutation. But given the earlier conversation about the funding environment, how do you pay for that? And is that different now than it would have been, not during the long trough that we're in?
Oh yeah, no, I mean, so 4 or 5 years ago, okay, yeah, and then you go back to where I was talking earlier. So 4 or 5 years ago, if we had demonstrated safety and efficacy, if we demonstrated safety and tolerability in healthy volunteers plus target engagement, I believe we would have probably been able to raise 100 to 150 million on that alone. That would have carried us easily through phase 2. Instead, with risk appetite being so nauseous, really, that we're taking little bites and more incremental steps because of the risk involved with 100 million versus 20. So can we do a half measure? It's not great for drug development. It's not ideal, but we've been making the best. We've been making the best of it. And I think what we've been able to achieve on very little funding is Herculean. If you think of the characterization we can now put forth for GT02287 to evidence of multiple biomarkers that are moving in correlation with what we believe is a clinical benefit, that's pretty good out of a phase 1B study. Now, again, we have to prove that up with statistics in the phase 2. But the totality of the signal that we're seeing among the patients in the phase 1B study is just impossible to ignore. So we think we would have been much more efficient 4 or 5 years ago. How we're going to finance it now is, we're talking with potential partners from large pharma, large biotech, mid-cap biotech, in fact, even too. We are agnostic to the type of, we just want to get this to patients in the most efficient way possible. Is that in our hands? Is that in the hands of a partner? Is that a co-development arrangement? We're happy to discuss all of that. We just don't want to lose any time. We just want to keep this drug on track so it gets out there as soon as possible, while we're hand-wringing over the right patients and the right way to go about doing things. We also have to focus on let's get this done. So we think by engaging with potential partners over the last couple of years that we continue to interface with them on a regular basis, we can come to some kind of an arrangement there. We're constantly evaluating capital markets and the opportunities to finance with more dilutive funding, or some combination of the two. Right now, we have enough capital on our balance sheet to finish the phase 1B study in the fourth quarter. We think most of the data from that study is in the can at this point. So we have very good understanding of the biomarker evidence of efficacy with GT02287. We're seeing some what we believe early signs of clinical benefit in these patients. Hopefully that remains durable over the course of the next 3 to 6 months while the phase 1b completes, but we're going to start the phase 2 study already.
That sounds good. So you mentioned before, I think, Gaucher's. There's also some other potential applications for this drug, dementia with Lewy bodies, Alzheimer's, and you have other assets too. How do you prioritize opportunities? Is it basically you just got to go all in to get to phase 2, do this phase 2 trial, or how do you keep these balls in the air?
Right. So due to Gaucher's also involving, that's a different patient population, lots of children in that patient population, a lot more clinical testing that needs to be done in order to dose in younger patient populations, those patients that are under 16. That was not a practical route for us to move in first. The most practical route to establish the efficacy of GT02287 was in Parkinson's, where there is nothing to treat, where the enzyme target is the same and very prominent in the disease process, and where we could show delta, clinical benefit with some reasonable time frame. So we chose to go there first, hoping we can get GT02287 approved on the back of that, and it becomes much easier to move it into other disease areas. For Gaucher's, dementia, Lewy bodies, Alzheimer's, we have presented data suggesting that GT02287 reduces tau in an Alzheimer's model. So we've got eyes on that as well. But for now, the decision on Parkinson's was the endpoint's very clear. It's part 2 on the MDS backed up by support from part 3 in that clinical scale for the FDA. The field is trying to validate biomarkers that are predictive of that clinical benefit so that drug development can happen faster. And the FDA, we're part of that effort with the biomarkers that we've discovered, moving in the right direction in association with GT02287. We feel like we're part of that effort, and maybe we can advance this biomarker forward for the field by continuing to pin it to a clinical benefit. And then the next sponsor who comes along who's able to demonstrate the impact on the same 2 biomarkers, maybe their lives will be a little bit easier than ours with the regulatory agencies. And that's fine. That's what we want to contribute to the field.
What's the average age of the patient in your Parkinson's trials compared with the average age of a Parkinson's patient in the population?
It's fairly similar. I believe the average age of the Parkinson's patients, I'm going to have to go back and check, was somewhere between 35 and 45 in our trial. So maybe a little bit on the earlier side. Typical Parkinson's patient is diagnosed in their 40s or 50s in most cases, but there are a lot of diagnoses in patients that are in their 70s. It's like this late onset type thing. It can be really, prominent folks have been diagnosed in their early 30s. So it's real wide spectrum, but they're typically elderly adults, well into their 40s and 50s.
So last main question is, you emphasize that Gain's focus on the mission and having truly disease-altering treatments going beyond the temporary symptom relief, incredibly important in Parkinson's in particular. So if the drug succeeds, and realizing we're a long way from that in terms of number of patients, trials, and time, but let's just take the good case.
We're getting closer.
Yeah, you're getting closer. I don't mean to. You don't need any, dissuasion. But for any patients out there listening to this, I want to give the impression this is not tomorrow. But if the drug succeeds, what would it mean for neurodegenerative diseases, including Parkinson's, but beyond Parkinson's too? What's the potential? What's the big kind of vision that you're shooting for?
If the safety and efficacy profile that we are in early days of seeing continues to play out, I think what we will have done is contributed an entirely new backbone. Let's just talk about Parkinson's for a minute. This will be the backbone therapy for Parkinson's patients until another disease-modifying therapy comes along that may be better or additive or just different. This will be a new backbone of Parkinson's patients. And with the tolerability, a lot of clinicians aren't going to care what our data looks like. They're going to give their patients a shot on it, regardless of their genetic subtype or whatever. That's just a fact in the clinical community. They'll say, well, using this in these patients.
Give it a shot.
Give it a shot. If you feel like garbage on it, we'll take you right off. But let's give it a shot. So I think what we'll wind up doing is taking GT02287. It will form the backbone of any kind of symptomology that still exists in the patients once we've caught them with GT02287. So hopefully we stop the progression of the symptoms where they are, but those symptoms will still be there. But the L-DOPA that they use, the other supportive medication that they use, the deep brain stimulation that they may be using, that stuff may be less relied upon. And that's great for patients, right? And then as the field gets more experience with the drug, they'll probably move it earlier, with or without our help with clinical studies, which we will do anyway. They will move it earlier and earlier into patients with the obvious objective of trying to halt their symptoms as soon as they see it. And if there is some, robust response to patients who have a GBA1 mutation that we uncover as we continue to develop and continue to evaluate GT02287, then there will be a case to be made to genetically test some patients to see what their status is to get them on before symptoms even occur.
Great. Well, that's it for another episode of the Health Biz Podcast. I'm David Williams, president of Health Business Group. My guest has been Gene Mack. He's president and CEO of Gain Therapeutics. If you like what you heard, please subscribe on your favorite podcast platform. And thank you, Gene.
Thank you, David. I appreciate your time.